everybody. In this video we're going to look at another genetics problem from chapter 7, in this case problem 7.12. This is similar to problem 7.11, so again if you listen to this video and you think you understand what's going on, you can go back and try problem 7.11 and see how things are going. It does mention in this particular problem that we're talking about two genes mentioned in that previous problem. So what I've written off to the side are just the two possibilities here. Basically, we're talking about two traits. So this is a dihybrid cross. The body of the fruit fly is either gray, which is wild type, or black, which is recessive. And the wing is either normal, which is wild type, or vestigial, sort of a small misshapen wing, that's recessive. The problem in the chapter suggests certain symbols that you could use here. And I'm going to suggest that we use whatever symbols we like. So in keeping with the Mendelian tradition, we can use a symbol like perhaps lowercase b for black body, and then the capital to indicate the wild type, the dominant allele in this case. So we'll use B for the body type. And for the wing, we can use maybe a small W for vestigial and a capital W for normal. I'm not using a V for vestigial here because in my handwriting I can't really tell the difference very well between a lowercase and an uppercase V, so I'll choose to use W. Doesn't matter what symbols you use as long as you tuck them off to the side here so that you know what's going on. Now they tell us in this problem that we have a phenotypically wild type female fruit fly. She's heterozygous for both genes. And so we could say off the top that our female is heterozygous for body type and for wing type. And she's crossed with a male that is a homozygous black vestigial. So homozygous black, homozygous vestigial. This would be a test cross, and that's why these sorts of problems are called two-point test crosses. We always have the experimental individual on one side and a completely recessive individual for the traits we're looking at on the other. Now, if this were Mendelian and we had our normal sort of a cross here, and we could diagram this out with a Punnett square, we would expect basically 1/4 in each of the four categories. We'd expect 1/4 that was black with normal, or sorry, gray with normal wings, 1/4 that was gray with vestigial wings, 1/4 that was black with normal wings, and 1/4 that was black with vestigial wings. That'd be our Mendelian expectation. Now when we look at the numbers that are given to us in this part of the problem, they tell us that we have 23 individuals in one class, 127 in another, 124 in a third class, and 26 in the fourth class. It's clearly not equal proportions. If they were close, if they looked kind of close to a 1:4 ratio for each one, then we could go ahead and do chi-squared and find out if really this does match Mendelian expectations. But we don't need statistics for this one. We have two groups that are clearly overrepresented, the 124, 127, and we have two groups that are clearly underrepresented. So this is definitely linkage. So I'm going to go ahead and write those numbers down, numbers of organisms. We'll write those here. So for our gray normal wings, if we read through, that was our 23. We have our next class is gray with vestigial wings, that was our 127. We've got black with normal wings, that was our 124 organisms, and black vestigial was 26. Now we know that our female that we were starting out with had a genotype that was dominant for both. Now that we're considering linkage, we don't want to just write her genotype as we have before. We'll want to write it in a linkage genotype. And so we'll consider where on the chromosomes those two alleles are. And what we know by looking at the data that's given to us, the more common cases, the ones that are overrepresented compared to Mendelian genetics, essentially these two classes, those are the ones that we need to look at as our parental types. And so one of our female's chromosomes had the dominant B and the recessive W, and the other one had the reverse. So we could write that here. And it doesn't ask us in this particular problem, but we would say this is a repulsion heterozygote because the wild type allele is on opposite pieces of DNA for each case. Now they say, do they indicate linkage? Yes, because we don't have Mendelian ratios. What is the frequency of recombination? In order to calculate frequency of recombination, we're going to take our number of recombinant offsprings out of the total, and then we can make it a percentage by multiplying by 100. So if the types that were indicated here in green were our parental types, that is, those combination of traits matched our parent, then the other ones, the underrepresented ones, will be our recombinant types. And so those are what we want to add up over here. Say 23 in one class and 26 in the other class, divided by the total. And in most of the problems in this book, the totals are nice numbers. So here, if we add up our 127, our 124, or 26, and 23, we get a total number of offsprings that were counted of 300. So then we go ahead and we do our math. I'll include this term here. Percent recombination is 23 plus 26, 49 out of 300. And that amounts to 16.3% recombination. Percent recombination is also for us the unit of map distance called a centimorgan. And that was sort of honorarily named after Thomas Hunt Morgan, who did a lot of the pioneering work with fruit flies. When they tell us to diagram the cross, showing the arrangement of the genetic markers, what we're really looking for is the bottom line. So here is the female's genetic organization. We know that she has dominant for body type on the same chromosome as the recessive for wing, and vice versa. She's that repulsion heterozygote. And the other part we'll need on our diagram of the cross is the distance, 16.3 centimorgans. And that is our final answer. From that, if we went on to do predictions or anything like that, we wouldn't use our Mendelian expectation of 25% in each case. We would say we'd expect the offspring to have 16.3% recombinant and the remaining 83 and change percent is parental. Once you've looked this one through, I encourage you to try problem 711, which is a similar gene organization. The numbers are different, which means that your results will vary slightly. Good luck.